fairgrounds, those are on Ganson Avenue?
I know vaguely where it is, but I can never remember the road over there. I know it's near downtown.
Ganson sticks in my head. That might be one.
I know they say it's going to come in the track, so
Yeah.
I'm very excited to be able to get it.
Well, I'm glad we opened up the eligibility now. So.
Yeah, but it's-
Get these to you because I found this.
They're right on the money. I don't have the downtown road names well enough yet. But I've been here for 4 years.
So Ganson's the one, so it's parallel to Lewis Glick, and between those two is the hospital. So if you go up Ganson, before you get to Cooper Street,
Mhm.
that's, I think, the area where the fairground is, yeah.
Next, we do bar pants at JSA.
Right, okay. Okay, Amanda, can you hear me all right?
Yeah, I can hear you.
How's the kitty?
He's good.
Happy Easter, kitty. All right, can you see the slides okay?
Yep.
And are the slides moving for you?
They are.
Okay, good. Okay. Hope you guys all had a great Easter. First. Would you like another online quiz?
Yeah, I like them.
You like the online quizzes? Let's do an online
I
quiz.
especially
then.
like how it's so much.
If you want to go the online route, that's good. Do it online. Okay, yeah, now I've got my bearings here.
Yeah, this is. I don't even know. This is so.
Okay, let's go ahead. Let's have a word of prayer and let's get started. Lord, we thank you for Easter. Thank you. We're back in the saddle. We pray now for our entity of projects. Watch over us, please. Thank you that you have healed those of us who are ill. Thank you that my daughter had a very mild case and that I didn't get sick. Now, Lord, please watch over us in Jesus' name. Amen. Okay. Projects. I'm stuff I need for the Western is speckled. And
So you're just gonna do it when it comes in?
well, yeah, they're being very cagey about when it's coming in. So, me no likey. So yeah, okay. Now, we ended up talking about the D-amino acid oxidase. And the D-amino acid oxidase exists in order to process the amino acids that come from the breakdown of the bacteria in our GI tract. So as you might guess, we get quite a few of these, and in the kidney, this enzyme is expressed in lots of places, not just the kidney. It's expressed at very high levels in the kidney. And I discovered, this is actually a slide taken from your book. I discovered there's a mistake here. This enzyme produces peroxide. So it's H2O2, so that's a goof. So we're going to There we go. Let's take a small look at this enzyme. It's actually quite interesting what this enzyme does. So the D-amino acid oxidase, it's abbreviated DAAO. It's a ubiquitous enzyme, and what this does is it takes the D amino acid and it will deaminate it. So you get the alpha keto acid. Just like that. And then this can be reaminated. And usually this will be something like glutamate. We'll give its group and you'll have alpha ketoglutarate typically, and then you'll get the L amino acid out of this. So you'll have so that's what this enzyme normally does, is it handles these D amino acids that come from bacteria. However, there's one other place where D amino acids make their appearance. So this is the way the enzyme normally works. This would be so it essentially gives you a substrate that you can convert back into an L amino acid, which is kind of handy to take the R amino acids, reconvert them into the D amino acids. Now this enzyme, because it makes peroxide, that should give you a hint where it is in the cell. Yeah, Dan, you're right. It's in the peroxisome. So it's a peroxisomal enzyme. Now, don't tell this to Aaron. This enzyme is not found in plants. But it's a pretty good enzyme that's used to mark peroxisomal extracts in animal cells. So typically in the kidney, as you saw, so kidney, it's expressed at high levels, very high levels. And in the kidney, we saw that it'll handle glycine by taking it'll take glycine and it will relieve this of the, so you get water and then you get peroxide plus the ammonium, and then from that you get the glyoxylate. So the other piece, you know what? I don't have this. Amanda can't see what I'm doing.
Yeah, I was just going to ask for notes from somebody if you didn't end up showing it to me.
Sorry,
Thank you.
there it is, okay. All right, so the peroxisomal enzyme, it's expressed at high levels in the kidney. It makes peroxide. Then the peroxide, remember, is dealt with by catalase. And catalase, very high levels of catalase in the peroxisome. So making peroxide in the peroxisome, no big deal. Lots of catalase to deal with that. And then from this, you make the glyoxylate. And then the glyoxylate then is converted into the oxalate. But Another place where this enzyme is expressed is actually in the brain. And here's where things get kind of interesting. So in the brain, you have NMDA receptors. Now, what does NMDA stand for? It stands for N-methyl D-aspartate. N-methyl D-aspartate. Now, this receptor typically will bind the neurotransmitter glutamate. So that's the main agonist of that receptor. However, another agonist of this receptor is D-serine. And this is an important regulatory agonist of this receptor. So one of the ways this receptor is modulated is by modulating levels of D-serine. And what enzyme do you think breaks down D-serine? The D-amino acid oxidase. So this enzyme ends up being very important in the brain because it breaks down D-serine. And by doing that, it regulates the activity of this glutamate receptor. And glutamate receptors play a very important role in several different functions, but also in several different pathologies. So because of that, small molecules that regulate this enzyme are now being looked at as potential psychiatric modulators. So I thought that was kind of interesting. All right, so let's get back to the slides now. So remember, take your pen and put 2 here. That should be peroxide. That's a mistake. All right, so you have on your handout the breakdown of amino acid tryptophan. It's on handout. It's page 6 on the thick one. And Amanda, those are all on one.
Thank you.
Okay. So tryptophan, the chain part of the tryptophan is taken off and converted to alanine. Then you have to deal with that ring. So how does one deal with tryptophan? So if you take the handout, you can go ahead and get a look at it. And this essentially will help us. So the first enzyme is a mixed function oxidase. This is tryptophan oxygenase. You can see it breaks down the first ring of the indole ring, and it makes the N-formyl, and then that molecule without that is called kynurenine. So it makes N-formyl kynurenine. And then kynurenine, and this is again a dioxygenase. N-formyl kynurenine, you might think that tetrahydrofolate would pick that up. In fact, it doesn't. It's just burned. You just put a water across that and it gives you essentially formate, and that's about it. And you degrade that to kynurene, and that is the enzyme kynurene formamidase. Kynurene formamidase. And now you have another oxygenase. This oxygenase, interestingly, is going to use reducing equivalents and molecular oxygen. And that's going to convert the kynurene to the hydroxylated derivative. There's a method to this. You'll see why that's the case. You might think it's breaking it down. Why do you want to put a hydroxyl group on there? You put a hydroxyl group on there, then if you take away those carbons, you make a carboxylic acid. That's why putting hydroxyl group there is very useful. Make the 3-hydroxy and kynurene. Now you're going to take off alanine and you use kynurenase. And this kynurenase is a pyridoxal phosphate containing enzyme. So that just lops the alanine straight off. And then you make the 3-hydroxy anthranilate. 3-hydroxy anthranilate. That's still an aromatic compound. B6 deficiencies will result in kynurene being excreted in the urine. When it hits the air in the urine, it turns a greenish yellow color. So your urine is a greenish yellow color. And typically a greenish yellow color could also mean you have a Pseudomonas infection. So sometimes this gets confused for a Pseudomonas infection of the bladder, when in fact it's a B6 deficiency. Okay, then you have 3-hydroxy anthranilate oxygenase, yet another oxygenase. That's going to open the ring and you're going to get a semi-aldehyde. A semi-aldehyde. The addition of water will cause the 2-amino 3-carboxymucinate 6 semialdehyde, otherwise known as ACS, to cyclize spontaneously. And that molecule is called quinolate. Quinolate is used in organisms that don't require dietary niacin to make their own niacin. So essentially, niacin gets made from the breakdown of tryptophan in organisms that make their own niacin. All right, so the next reaction is a decarboxylation reaction. And that produces the 2-amino mucinate semialdehyde, which you can see here. And then the 2-amino mucinate is converted to amino mucinate. You're going to oxidize that molecule. When you oxidize that molecule, you then make alpha keto adipate via reduction of the amino mucinate, and that gives you alpha keto adipate. Alpha keto adipate is going to be broken down by a pathway that will also be used to break down lysine. And the alpha keto adipate is converted to glutaryl coenzyme A by alpha ketoglutarate dehydrogenase. So you get glutaryl coenzyme A, and then your glutaryl coenzyme A, And so this enzyme, that's a Krebs cycle enzyme. And the glutaryl coenzyme A undergoes essentially the same reactions of beta oxidation to make crotonil coenzyme A, and then that just gets subjected to beta oxidation. You add a water across that, then thiolase splits it in half, and then you end up with two osteocoenzymes. So that's how you break down alpha ketoadipate and also tryptophan.
Does it break down the lysine or it undergoes the same thing that way?
Lysine gets broken down also to alpha ketoadipate. Then alpha ketoadipate gets degraded by first the Krebs cycle enzyme, then these enzymes here, those are just beta oxidation enzymes. Essentially, glutaryl coenzyme A gets degraded by the enzymes of beta oxidation. Now, tryptophan. It's important for us to know that tryptophan plays a very central role in the synthesis of two different neurotransmitters. So tryptophan, W, is used by the pineal gland to make melatonin. And then it's also used in the brain to make another extremely important neurotransmitter, and that's serotonin. So melatonin is made at night, and it has to do with our circadian rhythm. It inhibits the synthesis of other transmitters, neurotransmitters that are dependent, for which wakefulness is dependent. Serotonin is involved with so many different functions in your body. It's really remarkable how many things serotonin's involved with. But serotonin is made from tryptophan in really only 2 steps. So that's tryptophan. And let's get our double bonds in there. So the first reaction that's used to make serotonin from tryptophan is a hydroxylase reaction, and this is going to take tetrahydrobiopterin. Where do we see tetrahydrobiopterin before? Phenylalanine degradation, because phenylalanine gets converted to tyrosine. So that's a hydroxylation as well. So this molecular oxygen is going to come in, and then out of this, you're going to get water plus dihydrobiopterin. This enzyme is called tryptophan hydroxylase. And so this is going to produce tryptophan that has a hydroxyl group right there. So, oh, do I have room to draw this? Sorry?
You said this, you're drawing what it produces.
Yeah. So you make from this 5-hydroxy tryptophan, 5-hydroxy tryptophan. That's the first step in the synthesis of serotonin. All you need to do now to make serotonin is to get rid of this carboxyl group. And so that reaction is carried out by something called aromatic amino acid decarboxylase. This enzyme contains pyridoxal phosphate. So this enzyme is going to take the 5-hydroxy tryptophan. You'll get rid of a CO2 here, and you'll make from this and that's serotonin. So it's 2 steps to go from tryptophan to serotonin. Melatonin is also made in 2 steps, except melatonin synthesis is slightly more complicated. It's actually made from serotonin. So you have tryptophan at the start, so you have those 2 steps there. Then you have so you start out with serotonin, and then you are going to add to it an acetyl group. And the acetyl group is going to come from acetyl coenzyme A. That is going to attach to the amino group here. So that will give you 5-acetyl, sorry, N-acetyl 5-hydroxytryptophan. Is this on your handout? I'm wondering if I'm drawing this and you guys are going, gee, it's all here. I wonder why you're doing that. And now to this, you have a so if this is serotonin, serotonin sometimes is called 5-HT, 5-hydroxy tryptamine. So-called 5-HT. This is now N-acetyl 5-hydroxy tryptophan. This enzyme, by the way, is called 5-hydroxytryptamine N-acetyltransferase. So 5-HT, 5-hydroxytryptamine N-acetyltransferase. This molecule then is going to be methylated. It's going to be methylated right there. So you have a so-called hydroxyindole methyltransferase, and the substrate is S-adenosylmethionine. And from this, you get S-adenosylhomocysteine. And now this is so-called hydroxyindole methyltransferase. And that gives you melatonin. And melatonin is pretty simply serotonin with the acetyl group plus a methyl group over here. SAM,
Okay, so it's an A.
S-adenosylmethionine and S-adenosylhomocysteine. And that is melatonin. So 2 steps from tryptophan to serotonin, and then 2 steps from serotonin to melatonin. While we're on the subject of neurotransmitters, so glutamate, which is itself a neurotransmitter, that's glutamate, single letter amino acid code E, that can be decarboxylated. So this is going to essentially leave, that one there will end up leaving. This is glutamate decarboxylase. Glutamate decarboxylase is a pyridoxal phosphate containing enzyme. And from this, you're going to get this is gamma amino 1,2,3,4 butyrate. Otherwise known as GABA. GABA is the main negative or inhibitory neurotransmitter in the nervous system. And it's made from glutamate, which is an excitatory neurotransmitter. Okay. Enough of that. Let's go back to our slides. All right, so remember, and this should maybe help refresh your memory that this step here utilize tetrahydrobiopterin. So phenylene hydroxylase, remember, mutations in that cause phenylketonuria, and then mutations in either tyrosine aminotransferase or the p-hydroxyphenylmethyl dioxygenase will cause tyrosinemia. Then mutations over here in fumarate acetoastase, they can cause tyrosinemia. 3 different types of tyrosinemia. And this group of mutations, tyrosinemia plus PKU, they're called hyperphenylalaniemias. Hyperphenylalaniemias. These are due to loss of function mutations in the genes that encode those enzymes. So the inability to degrade phenylalanine to tyrosine increases serum levels of phenylalanine that will interfere with the synthesis of particular neurotransmitters. When you interfere with the synthesis of particular neurotransmitters, you can retard development of the brain. When you retard development of the brain, you end up with someone who is mentally challenged. And that mental challenge, of course, can be mitigated by reducing the amount of phenylalanine in the diet. So if you reduce the amount of phenylalanine in the diet early in life, after the blood-brain barrier gets formed so that phenylalanine just doesn't make its way into the brain, you can spare them the developmental defects. And as we said before, this is one of the panel of genetic tests that you get in certain countries. Not so much in this country, in Scandinavian countries where this one's prevalent, is definitely the case. And you can order it in the United States if it runs in your family. You can order these tests for hyperphenylalaninemia. You would have to reduce certain foods that are rich in phenylalanine. It's a very restrictive diet. Your protein sources have to be mainly from plant sources. Even then, you have to be very careful which plant sources. So
How long do you have to continue the diet until you see results?
if you don't stay on the diet, you'll tend to get sick because the end products will build up in your blood and will cause symptoms. So it's most crucial at those earliest stages in life. These kids, they can't breastfeed because breast milk has a lot of phenylalanine. So it's not an easy life, as you might guess. And patients with tyrosinemia will show early symptoms at least similar to those with PKU. That's why tyrosinemias, phenylketonurias are all collectively called hyperphenylalaniemias. And then mutations in the homogentisate 1,2-dioxygenase gene will cause alkaptonuria. That does not cause mental retardation, but it causes the dark urine and the late onset arthritis. The dark urine comes from homogentisate being excreted in the urine. Now, albinism results from the absence of melanin synthesis in the skin. Melanin is also made from tyrosine. So tyrosine is broken down in the skin by tyrosinase, and that produces something called DOPA, dihydroxyphenylalanine. DOPA gets further oxidized by tyrosinase to form something called a 3,4-quinone, sometimes known as a dopaquinone. 3,4-quinone is extremely unstable, especially in light. and it cross-links multiple times and it forms this polymer. And that polymer essentially is what's responsible for the pigment in your skin that you get when you go suntanning. That is due essentially to cross-linking of tyrosine. And so some models have to have a certain amount of sun time every day because they need to have that kind of golden look. Some women just can't get it, depending on your genetics. Some just have it naturally. It's called life. It's not fair. But yeah, if they do that, some of them will follow diets that are rich in tyrosine so that they have more tyrosine to cross-link. They get dark faster. They don't have to spend as long in the sun. And unfortunately, some of those ladies have been subject to, I think male models do this too, are subject to certain quackery diets, that the sun tanning diet, so on and so forth.
Does it actually work?
You can feed people tyrosine supplements. They'll tan noticeably darker, quicker than other people. So yeah, it's just whether or not,
Is it a healthy thing for average people to do?
if you eat extra tyrosine in your diet, it's not going to hurt you. Tyrosine is mainly found. My guess is you have a little trouble getting dark.
I do, yeah.
Yeah, given your genetic heritage. That's my guess. You're probably very Northern European. And my middle daughter, Stacy, has the same trouble. Her Dutch blood runs very thick. Emily, on the other hand, no trouble. She's got my Mediterranean heritage. I get very dark in the sun, but that's my Mediterranean heritage. My wife actually was very Dutch, tans actually quite well, but she burns more easily than I do.
Yeah.
Rachel tans really easily. Stacy burns. Stacy's like you. So,
As I'm getting older, I burn a little less, but I'm also just a little bit.
yeah, yeah,
It's awful. but
yeah,
all
yeah. There it goes. Tyrosine is a major substrate for the synthesis of some extremely important neurotransmitters. And of these, you have various catecholamine neurotransmitters, of which epinephrine and norepinephrine and dopamine are the most important. So tyrosine hydroxylase will convert tyrosine into dopa. And dopa, as we said before, is this dihydroxyphenylalanine. And dopa is the direct precursor for the synthesis of dopamine. And then dopamine is a precursor for the synthesis of epinephrine, norepinephrine. And then norepinephrine is the precursor for the synthesis of epinephrine. So tyrosinase that makes melanin is only found in melanocytes. That's in the skin. The tyrosine hydroxylase that's used to make neurotransmitters, that's a slightly different enzyme that's found in other tissues like neurons and things like that. Now, having said that, melanin is found in other places in your body. In your midbrain, you have something called the substantia nigra. The substantia nigra has melanin in it. It needs that melanin to be able to do its job. And that makes melanin too. But the main makers of melanin are in your skin. Oh, let's talk about these neurotransmitters because that's quite important. All right, so well, I'm terrible at drawing these benzene rings. Okay, that's tyrosine. Single letter code Y. Camera didn't switch over? On you. Come on. Okay. There. So that's tyrosine. Tyrosine hydroxylase. We'll use once again tetrahydrobiopterin. And you'll get from this dihydro and then also oxygen, and then also here water. And now you're going to hydroxylate this right here. That's going to give you dopa. No other part of the molecule is changed, just that. So this is tyrosine hydroxylase. This is dopa. Now, if you get rid of this, you're going to make dopamine, and the enzyme that does that is the same enzyme that was involved in the synthesis of serotonin, the aromatic amino acid decarboxylase. So aromatic amino acid decarboxylase, and you get dopamine. Huh, that's ugly. That's dopamine. And dopaminergic neurons will typically have high levels of tyrosine hydroxylase. So that's dopamine. I'm kidding. Am I out of paper? So that dopamine then is the precursor for the synthesis of norepinephrine. Now, to make norepinephrine, all you need to do is put a hydroxyl group right here. So this, you're going to actually use vitamin C as a substrate for this. So you're going to use ascorbate. And then once again, molecular oxygen. And you'll make dihydro. Ascorbate plus water. And you'll now make, oh, applause, please. This is called dopamine beta hydroxylase. Dopamine beta hydroxylase. This is probably one of the reasons why scurvy is not only accompanied by loose teeth and loose connective tissue, but also by mental breakdown as well. Because you can't make this, and this is norepinephrine. Commercially, this is available in the hospital under a product called Levofed. That's used for shock and that sort of thing. And then in order to make epinephrine, just guess what you have to do to make epinephrine based on what you saw before. No, you're at a methyl group here. And you're now going to end up with a methyl group here. That's epinephrine. Oh man, I'm getting better. And this is called phenylethanolamine and methyltransferase. Phenylethanolamine and methyltransferase. And this is epinephrine, otherwise known as adrenaline. So all three of these from epinephrine, norepinephrine, and dopamine are all vitally important neurotransmitters, and they all start out being made from tyrosine. There's this guy in the gym today. He had this pre-workout punch he was drinking, and I'm terribly nosy. I grabbed it and I looked at the ingredients. There was 1,000 grams per serving of tyrosine in it. And I said, I'll betcha it's so that you can make these neurotransmitters that are part of the sympathetic nervous system to jack you up. That's probably the strategy behind it. I doubt you're going to get a ton of adrenaline because that's got to cross the blood-brain barrier. The amount that crosses the blood-brain barrier is highly regulated. But hey, it's good marketing. So, all right. No, that's fine. Come on. Come on, computer. You can do it. Okay. Now, isoleucine and valine are both converted to succinyl coenzyme A, but so is methionine. And methionine degradation is it's not one of these. I don't see it. I'm sorry. Anyway, so we covered the branched chain amino acids already. But first of all, methionine participates in methylation reactions. So that converts it to homocysteine. So you have a series of reactions that allow it to make S-adenosylmethionine. So you have the methionine adenosyl transferase that uses ATP and methionine to make S-adenosylmethionine. Then you have your methyltransferases that makes S-adenosyl homocysteine. Then you have an S-adenosyl homocysteine hydrolase. Now you get homocysteine and adenosine. And then homocysteine is condensed with serine. And by being condensed with serine, you make this intermediate called cystathionine. Cystathionine. And that is catalyzed by cystathionine beta synthase. Cystathionine beta synthase. You can see this here. Serine is going to now, so here is your, well, here's your homocysteine, here's your serine, right here. So you can see essentially you lose the hydroxyl group, you lose the water. So this hydrogen goes to the hydroxyl group, you lose the water to dehydration, you form the bioether, essentially. And then from this, you're going to make cysteine. So you have cystathionine synthetase, and then you're going to hydrolyze cystathionine via, it's either called cystathionase or cystathionine lyase. It's called both. That gives you cysteine and alpha ketobutyrate. And alpha ketobutyrate is subjected to alpha ketoglutarate dehydrogenase, which gives you propionyl coenzyme. And the propionyl coenzyme goes through the same cycle as beta oxidation. And that's shown here. And then the valine, isoleucine, leucine, we've covered that already. Just to remind you, essentially, you have a large complex, the complex branch chain alpha keto acid dehydrogenase complex. That complex handles those, converts them into their alpha keto acids, and then these undergo a process that kind of sort of resembles beta oxidation, and that is on this handout. And then the other reaction. Yeah. Well, we went over that already. So, and the products, as we said before, they're relatively unsurprising. Propionyl coenzyme A, succinyl coenzyme A, so on and so forth. And then leucine is unusual because it's converted to acetoacetyl A and acetyl A. And mutations in the branch chain alpha keto acid dehydrogenase complex will cause the maple syrup urine disease, sometimes called branch chain ketoacidosis. But you get, it's inherited, and you get the vomiting, convulsions, early death, so on and so forth. All right. Okay, I think that does it for degradation of amino acids. I'm trying to think if any got left out. Well, then it's a good place to stop. Next time, then, we will cover synthesis. Remember, you're only responsible for synthetic pathways that occur in humans. Otherwise, you just need to know the families. So what goes in, what comes out.
degradation.
and urea cycle,
Oh,
and-
urea.
yeah.
Are you going to include the biosynthesis that we're still learning then? I think the syllabus said it should be like the exam is. Sometime in the 20s of April to like not next week. But the week after.
I'll probably include all the amino acid stuff.
So amino acid. in here.
Right. I probably won't include the nucleotide biosynthesis. Which, by the way, those pathways will be the worst pathways you'll see.
How did you study for these, like when you learned them the first time?
Kept drawing them out, drawing them out, drawing them out, drawing them out.
So, like, if there's a lot of drawing, how can we expect the exam to look like? Are it still gonna be multiple choice?
Yeah, it'll still be mostly multiple choice, but I'll still ask you about various steps. And if you draw them out, you'll know the steps.
Right.
So
Then photosynthesis.
that was on the last exam, isn't it?
I don't know. I think we may have taken it off.
No, I don't think it was. I don't think it was. So photosynthesis as well. So yeah, there'll be plenty for the next exam.
Out of curiosity, for our article summary, what's coming up? I think all of us are a little behind tonight. We asked for an extension on it.
Sure.
Sorry. Cool.
Okay. Amanda already signed off. Bright-eyed and bushy-tailed.